Okay, good afternoon, everybody. Welcome to Abhisha Thayambath, which is a graduate student in the physics department at North Carolina State. And she's working with Julio Belmonte. And her research uses the cellular POTS model to study tissue level polarity patterns and examine the role of cell mechanic properties during morphogenesis. So some basic questions in development and morphogenesis. And please, the floor is all yours for giving your presentation. Thank you.
Thank you, Lorenzo, for the kind introduction. So I'm going to talk about subcellular POTS model for planar cell polarity without morphogens. So, okay. We know that cells are not homogeneous bags of fluid. Most cells have a heterogeneous structure and there's spatial differences in shape and function. So this is typically referred to as cell polarity. A classic example of a polarized cell are epithelial cells with apical basal polarity, where apical membrane facing outside surface of the body or the lumen of internal cavities, and the basal membrane orienting away from the lumen or outer body surface. So these cells also exhibit something called planar cell polarity, where specific proteins called PCP proteins localize to the apical junctions and distribute themselves asymmetrically to the opposite poles of a cell. So this polarization of apical cells, epithelial cells, are orthogonal to the apical basal axis, and hence it's called planar cell polarity. This is mostly found in external structures such as body hairs, sensory hairs, wing hair, etc., in both vertebrate and invertebrate epithelia. So cells orient themselves and their neighbors by establishing an asymmetric expression of PCP proteins on neighboring cells. So now, the underlying pathway comprises different levels. So at the molecular level, there are core proteins that form intercellular complexes, and then there are local feedback interactions between neighboring cells that stabilize complexes of the same orientation. So these are the proteins involved in the PCP pathway. So you have flamingo, frizzled, Van Gogh, prickle, Diego disheveled. So these proteins form intercellular complexes, and then local feedback interactions are stabilized. So in Drosophila wing, this leads to the formation of wing hair from the distal end of each cell. So these hexagonally packed cells at a tissue level orient along the proximal distal axis, which is basically from left to right, with the help of some global cues. So starting from a configuration where all these proteins are uniformly randomly distributed, over time the cells, the proteins segregate and they localize to opposite ends of the cell. So now, various models have been proposed to explain the mechanisms behind planar cell polarity, and these models try to clarify how the intricate interactions among PCP proteins lead to the observed orientation patterns. So understanding the mechanisms involving PCP there are various modeling approaches. So first are explicit signaling models that try to consider the molecular level processes explicitly. So these models delve into the intricate molecular processes. They try to analyze the protein interactions and subcellular localization quantitatively. Then there is a second class of models called phenomenological or reduced models that try to capture the essential dynamical features of PCP. So these models capture the coarse grain behavior of the system and highlights the core principles that drives planar cell polarity. So here is an example of an explicit signaling model. So this is a reaction diffusion partial differential equation based model and simulation that fully reproduce PCP phenotypes. So the geometry involved in this model is basically hexagonally packed cells that are fixed in the lattice. Then you have movement of proteins and complexes within each cell that is modeled as diffusion. Then there are feedback loops that arise from interactions of PCP proteins within and between cells. and that is sufficient for the local alignment of PCP complexes between small groups of cells. However, there is a global biasing signal that is required to align the orientation along proximal distal axis. So this model reproduces the known PCP phenotypes and establishes the feasibility of the biological model based on feedback interactions between proteins that are localized inside each cell. However, the model assumes some sort of a pre-pattern and does not explain how each cell in the tissue initially becomes polarized. A second example of an explicit signaling model is here. So the motivation for this model, the asymmetric Flamingo bridge model, is the hypothesized presence of a gradient that provides the initial asymmetry to all the cells, which is later amplified by feedback interactions. So this model is pixel-based, so cells are pixelated, and reactions take place at each pixel. So it could be either among molecules present in the same pixel of a membrane or in adjacent pixels of different cells. So this model is different from the previous model because of certain intricate details of molecular bonding between the proteins. Also, this model is again a reaction diffusion model where you have hexagonal cells represented on a 2D lattice. So starting from an initial configuration where all these PCP proteins are uniformly distributed inside each membrane, the molecules segregate to proximal and distal boundaries over time. So the model successfully reproduces the wild type as well as mutant PCP phenotypes and is comparable to the previous model. Although the model is it's stochastic compared to the previous model, the authors claim that it's robust with respect to fluctuations in initial gradient, cell shape, etc. The downside of the model is that it fails to account for local orientation in the absence of any one of the core proteins and to consider other cellular level processes like cell divisions, etc. during development. Now, let me switch gears to the second class of models, which is basically phenomenological or reduced models. And here is an example inspired by the conceptual resemblance to the patterning of electron spins in a ferromagnet or an Ising type lattice model. So this model basically obeys a simple short range consensus rule to simulate what seems to be a self-organized process. So you basically start with a set of randomly oriented vectors, and then the orientation of each vector is modified by adding to it the vector sum of its 18 closest neighbors after that sum has been scaled by certain percent. So this is basically a simple cellular automata model in which a local consensus rule acts iteratively to bias each hair's orientation in favor of the average orientation of its neighbors. So they describe two systems for hair orientation. So there is a global orienting system that acts early in the development, and then there is a local self-organizing system that acts later. So the authors consider three different scenarios. First being the time evolution of a 2D lattice of hair follicles from a randomly oriented initial configuration without any global bias. So you can see from this figure that for this case, there are swirling patterns generated over time. So which is a PCP mutant pattern. The second configuration is again a random initial configuration, but with a global bias that would help the follicles to align from left to right or proximal to distal direction. They also examined a third configuration where there are a few rows in the initial configuration that are perfectly oriented from left to right, and then over time, the rows that were initially oriented helps the rest of the rows to polarize from left to right, and it's highly resistant to perturbations from other follicle alignments. However, the authors do not explain how robust or sensitive the model is when the size of the lattice is increased. So the maximum system size evaluated in the model is like 400 hair follicles. But since mammalian tissues are comprised of hundreds and thousands of cells, we need some model that would examine how robust it is with respect to system size. So Another important work in this reduced or phenomenological models is this. So here, the mechanisms that globally orient planar polarity are investigated using a combination of experiment and theory. So according to the authors, proximal distal orientation of PCB complexes does not arise from a random initial configuration, but rather it evolves from a global polarity pattern present at the pre-pupil to pupil transition in Drosophila wing. So at pupil stages, the wing hinge contracts and the wing blade elongates along proximal to distal axis, as you can see here in the figure. So this process exerts a mechanical stress leading to cell flow patterns that would help polarization. So basically, polarity that was already established when the tissue was small can be propagated during development. So authors also claim that there is no need of a boundary signal or some sort of a gradient to act over long distances directly. So they model this using a vertex model based on attractive interactions between the proximal and distal PCP domains across cell boundaries and repulsive interactions within these domains inside cells. So this model takes into account cell mechanics, cell adhesion, and cell division, and they do not attempt to describe all details of interactions between PCB proteins. So to summarize, only a few models in these categories attempt to describe the reaction between PCB molecules, and most of the subsequent models are simplifications. And most models use some sort of a tissue gradient or some persistent global signal. And only a few models investigated cell growth and division. Most importantly, many of these models were static, and they don't have a metric for global polarization, or they did not investigate the robustness of polarity with increasing system size or the number of cells. So to summarize the biology, the most defining feature of planar cell polarity is the alignment that is robust across the tissue axis that suggests that there is some sort of global directional cue that helps the cells do this. However, we still don't have a clear understanding of what these global cues are. Potential candidates being tissue level gradients or mechanical forces. So current understanding shows that there is a lack of evidence that supports the requirement of wind gradients or wind ligands for PCP in Drosophila wing. And there is this recent paper in 2012 paper that suggests that morphogen gradients orient PCP vectors through their effects on growth and morphogenesis. So in a sense, cell growth and division from a pre-polarized domain can better help the cells achieve long-range polarity, and that could be a better strategy. So using these ideas, we are going to use cellular POTS model, basically CC3D, to develop a model for planar cell polarity. So why do we need a CC3D based model? So it's very easy to represent the spatial distribution of PCP proteins, and CC3D's framework integrates dynamic compartments that enables a comprehensive representation of heterogeneities inside each cell. So it's also easy to integrate essential cellular processes like cell growth, division, cell shape changes, and cell rearrangements. So as you know, in cellular POTS model, a generalized cell is a domain of sites on a cell lattice that share a common index, and this can represent biological cells or subcellular compartments, clusters of cells, etc. And then you have an effective energy that defines cell or domain properties and behaviors such as size, aspect ratios, adhesion preferences, etc. And the dynamics is implemented through Metropolis algorithm. Now, how do we use dynamic compartments to develop a model for PCP? So using dynamic compartments, a cell can be modeled as a collection of objects. And these compartments can represent either the physical structure of a cell, like nucleus, cytoplasm, and cell membrane, or abstract regions inside a cell, so which could be apical, lateral, and basal polarity of an epithelial cell. So this will be covered more in depth on module 6.1 and 7.2, I hope. but I'm going to use this idea to develop a model for PCP. So in our model, these dynamic compartments represent the internal distribution of PCP proteins inside each cell. So each cell has 4 domains: cytoplasm, proximal, distal, and lateral compartments. So each of these compartments, for example, proximal and distal compartment, represents the PCP proteins that gets localized to these regions of a cell. So in the absence of a neighbor or any interacting external environment, these domains are mixed inside each cell. Now, over time, when you have a whole bunch of cells, the system self-organizes, and starting with a completely uniform random distribution of these proteins, over time, the system self-organizes using addition energies alone. So we set the external addition energy, the contact energy between proximal and distal compartments, to be very low, and the internal energy to be very high, so that they segregate inside each cell to opposite ends. So we could also generate the mutant PCP phenotypes using our model. So a frizzled mutant phenotype is observed when you disturb the frizzle compartment here. So we see that polarity is disrupted at the distal end of the tissue when you introduce a frizzled mutant. And if you introduce a Van Gogh mutant, polarity is disrupted at the proximal end, which is similar to what you see in experiments. Now, in order to quantify the amount of polarization, we used a metric, which is basically the order parameter used by the flocking model community to describe the collective movement of self-propelled particles. So it's basically the average of the normalized distance between proximal and distal compartment center of masses over the entire lattice. So you can so phi is a global polarization metric, which ranges from 0 to 1, 1 representing a fully ordered system, 0 representing a completely disordered system. Now, we tried to understand how different kinds of boundary conditions can affect the long-range polarity of the system. We started with the simplest case, where we have periodic boundary on all sides and there are equal number of cells along both the directions. And then we try to increase the system size and see how robust is the polarity. Similarly, we also examined a second boundary condition. A second boundary condition where you have a left boundary orienting signal, and that helps the cells to orient along proximal to distal direction. And if we compare the results from these two cases, we can see that from the first graph, if we plot the number of cells versus the global polarization metric, for the periodic boundary case, as we increase the number of cells, the polarization decreases. Similarly, for the boundary orienting signal case as well. But the configuration with a left boundary is still better compared to a periodic boundary condition. And also in periodic boundary case, the cells are free to align in any direction. So yeah. Also, the time taken, average first passage time, is the time taken for the system to reach 0.9 polarization, which is higher for the boundary left boundary configuration when compared to the periodic boundary configuration. But either way, we see that as we increase the number of cells in the system, the polarization decreases, which means just a boundary orienting signal is not sufficient to generate long-range polarization. So what happens if we try to increase the number of cells, more cells, if there are more cells away from the boundary? For example, you fix the number of rows, and then you increase the number of cells along towards the right side. How robust is the global polarization as there are more cells further and further away from the boundary? You can clearly see that this is even worse compared to the previous case. So before, at least we could have a 0.8 as the global polarization. But if you have a system like this, you can see that it drops below to even 0.5 as you have roughly 900 cells. So we need something that could fix this. So we tried to incorporate cell growth and division into our model. So basically, we started with the case where you have a pre-polarized patch of tissue, and then you let the cells that are at the boundary, that are in contact with the medium, to grow and divide. And cells repolarize after each division. So during the division, just it loses all the polarity information. So the cells becomes pixelated as in the initial configuration. And then they look at the neighbors and then readjust their polarity. So if we calibrate the time scale for cell division and the time it takes for the cells to orient with respect to its neighbors, we can attain a better result. So this is implemented using mitosis steppable in COMSOL3D. So we found that this works for larger systems, and it helps in achieving polarization faster than starting PCP signaling all at once. And this works in the absence of a long-range global morphogen. So if you can see the graph, you can clearly see that for the case when you don't have cell proliferation, the results are really bad as you increase the system size. When the system size is, say, 900 cells or 1,000 cells, the global polarization drops down to 0.5. Whereas if you start with a small patch of tissue that's already polarized, and then you let the cells at the boundary to grow and divide, you can see that the polarity propagates within the entire tissue, and it works even for very large system sizes. But there is a problem. So if we consider, why do we let the cells only on the boundary to proliferate? What if we let all the cells along the tissue to grow and divide? So basically two cases: cells on the boundary alone undergoes division, a second case where they are uniformly dividing. So which one do you think would work better?
Anyone? Erin, Sharon?
The second one? The second
Yeah.
one?
Yeah, the second one works. There is a problem with the first one. The problem being, yeah, you can see that at the beginnings, it works perfectly, but later when the tissue size increases, if there is one cell that doesn't behave properly, it affects the polarity of all the cells, and gradually there is a misalignment. So all the cells either points towards up or towards down. So this happens mostly because when you divide the cells that are at the boundary, it sees only half of it sees the polarity. But instead, if you have a entire tissue and you pick a cell in the middle of the tissue, it sees the direction from all the neighboring cells. So we think this could be the reason why there is a polarity misalignment for this case, and not when you divide cells randomly from a big tissue. So yeah, uniform cell proliferation with a boundary signal kind of rescues the global alignment direction. But in either way, the idea that if you start with a small patch of tissue and then you let the cells grow and divide, the polarity propagates, even though there is a slight misalignment for the case when you do it only for the cells along the boundary. So we could also incorporate other cellular processes in our model. Basically, if there are processes where PCP acts upstream, for example, you can do cell elongation using the direction specified by PCP. We do this using the focal point plasticity plugin. So basically, you make a link between the two compartments and you set the target length of these links and the direction determined by PCP. Similarly, we can also do cell migration with direction specified by PCP. And we use the external potential plugin to do this. And finally, we use the PCP direction to simulate mediolateral intercalation during gastrulation in amniotes. So there is this process where a primitive streak is formed when cells undergo convergent extension movements. So to do this, we first let the primitive streak cells polarize along the, say, perpendicular direction. and the cells would pull each other with a force perpendicular to the PCP axis, leading to convergent extension and the formation of primitive streak. So we can use the PCP model that we developed to for all these processes as well. We can incorporate other cellular processes, also use it for primitive streak formation. And we could also simulate apical basal as well as planar cell polarities together using a 3D model of this. And yeah, that's pretty much it. And thank you. Are there any questions? I think I finished too early.
Worries at all. It was very interesting. Yari, Charles, Gilberto, do you have any question?
No, I'm okay.
If no one has, we'll go with one because I might have missed it, but your polarization, if you want to expand on that, okay, you start from mixed, but then the compartment in your cell, what they follow, they have differential adhesion.
Yeah, they do differential addition. Yeah.
Okay, so it's like a cell sorting simulation, but with pixel in a cell.
Yeah.
Which really are cells because this is a compartment. So each of the compartment, how do you size that? Is a diffuse cells or is the one voxel, one cell?
As in, can you repeat the question?
I see that when they are pixelated, the cell looks like one cell is one pixel, one voxel.
Okay. So you're saying how do we
Right.
assign the fractions of each compartments? We kind of fixed a ratio, as in 20% should be the proximal and the rest should be distal, like that.
Okay.
But I think we can experiment with that and see how that would affect the polarity.
Nice. Okay. Well, I might have more, but on the biological side, I'm very interested in endothelial cells. And I know that they polarize, but one way they polarize, and you mentioned perhaps at the beginning of your talk, is because of mechanical signaling. Actually, shear stress, which set a vector force, so there is a direction. And this, it appears that is now might be mediated by mechanotransduction by either notch. Probably there is PCP involved, but yeah, just do you want to expand a little bit more? If can you introduce mechanical forces in your simulation where they become like the
Yeah, that's very ambitious because the Drosophila wing is like huge, and I think I have a slide about how it actually happens there. So you have these radial directions at the beginning, and then these cells flow towards the center, and that's how it aligns. But it's I feel like it's way too ambitious for me to do it, simulate that, and yeah.
I was thinking more perhaps simpler forces that are homogeneous across one direction. Since like I'm thinking like flow in a blood vessel, you have a flat surface. Let's say in a dish, you have endothelial cell growing in a dish, and you can make the flow going across the cell layer. That might be something you could apply in CompuCell, like as a force that is a kind of homogeneous across your tissue, and could that be linked to your already wired system? Just a curiosity, obviously.
Yeah, that's very interesting. I think, yeah, we'll have to think about it, and yeah.
Okay, well, that was all that I have to ask. Let me see. I see something in a chat. I'm afraid I didn't sorry not to ask a good question, but I'm sure we're reading the paper. Well, I guess this will come later on. But I think this is a very interesting work that you have done so far.
Thank you.
Okay. Well, if nobody has any further question, I guess we can well, stop the recording.